Okay. That's life.
Time's up.
Right. All right. Okay. So I moved to UCR. And so just to show the difference between my usual environment, is how it used to look like. And then that's UCR. You recognize that part, right? So it's getting to minus 30 now in South Bend, Indiana. So I'm going to talk about several subjects. But basically, the focus will be on multi-scale modeling and experimentation, and what modeling can help, well, tell us about specific mechanisms in biology, and what physics has to do with it. So the first example is Myxococcus hantos, or it's called myxobacteria. It lives everywhere. You can go outside and find this bacteria. And this bacteria behaves in a different way when it has enough nutrient and when it's starving. Okay, or being starved. So before I get into details about this bacteria, I'll just explain why it's important to study bacteria. Well, first of all, people study bacteria because they want to know more about embryo development, right? So who are we? We're human, right? Living beings, we consist of millions, billions of cells of different type, and these cells form tissue, they form organs, and there's a hierarchical structure, but we're just a bunch of cells. Okay, when I was doing, say, mathematical physics, life was so simple. Now I can sleep at night. Okay, what if something goes wrong with one of these cells and so on? So we shouldn't be worried that much because there are a lot of local controls. If something goes wrong and cells mutate all the time, unfortunately, everybody sitting in this room has cancer in the sense that cells constantly mutate, but they get killed by their neighbors, actually. There is immune system response, and actually there are local responses, and we just need to kind of appreciate that. But in order to learn how cells build multicellular kind of populations, people look at bacteria, because this is how life started on Earth, right? And I came from Notre Dame. There are other ideas, okay? But I insist this is how it started. That's one reason for studying bacteria. Second reason is it's becoming a threat. So have you heard about antibiotics? No. Right. I have 2 kids. I know all about it. So bacteria are becoming drug resistant. So my second example of Pseudomonas is the main infection in hospitals. We all have it. But then they're becoming aggravated because patients are treated with a whole bunch of different types of poisons, let's call them this way, antibiotics. And bacteria mutates and finds a way to become even more deadly. Okay? So that's another reason for studying it. And then we're full of bacteria. We have bacteria in our gut. We won't be able to digest food without bacteria. So there is a symbiotic relation between humans and bacteria. And guess what? Bacteria are tough. God forbid something goes wrong, they're going to start it from scratch. They're going to start figuring it out. They're going to become social probably, and maybe in 1 billion years, another human is going to appear. So let's be careful. But the bacteria can survive nuclear disaster, by the way. Okay, so enough. I think I have your attention, right? For whatever reason. So this bacteria is amazing because when they have a lot of food, they just swarm. Swarming, it's like swarming bees, right? They kind of move around, explore the environment, and the whole population expands because they keep dividing. And they need to move out, otherwise they deplete all the nutrient and they're going to die. They're social. They cannot survive on their own. Each bacteria looks like a small submarine. They have different motility mechanisms. They're self-propelled. Okay? At one end, they have this pili 4, which they shoot one at a time in a cone. And these pili get attached to other cells. Pili do not get attached to the substrate. Okay? So it's kind of the cells constantly touching where other cells are and where they're going. So pili gets attached to a cell, and there's a motor inside of bacteria. It pulls, and bacteria kind of moves a little bit in the direction of the motion of that cell. Release, they shoot another pili in the cone, another cell, and so on. It's a social mechanism. The other engine is called slime production. It's covered in slime. It's not quite clear how they move. Some people think they kind of go through a spiral motion by kind of producing slime from different jets on the surface. So people are kind of looking into, and there is a group at Berkeley, they came up with that idea. The important thing is that each bacteria has two motility mechanisms, and they're the reverse. And I'll run simulations. Not simulations, these are experiments. And I don't know what's going on. But we'll just move forward. So you're looking at the edge of the swarm population of bacteria. And this bacteria kind of is very active. The whole movie is speeded up. For some reason, I'm unable to show it. They form like spirals, and then they kind of go outside. They produce slime. They lay down slime tracks, and other bacteria start following the lead. It's easier for them to move along slime tracks. Nobody knows why. But the important thing, that was this movie, they start oscillating. Each bacteria oscillates. On the average, every 10, 12 minutes, it stops and starts moving in the opposite direction. It's not just stops. It cuts pili at one end, regrow them on the opposite end, rearrange the slime production. Takes a lot of energy. Nothing in biology happens without reason. So this reversal was kind of an open question in microbiology for a long, long time. And in order to test certain or produce certain hypotheses, I think I'm being tested. So it's like it's kind of flushing all the time. And it was raining all day. So anyway, we came up with a simplified model. I'll explain what it is in a second. And we tried to answer why they reverse, why it's beneficial for the whole population. Okay, so each bacteria in the model is represented by a certain number of nodes connected by springs. And this is a Hamiltonian describing energy of the system. So bacteria can stretch and it can bend. And there is a stretching and bending energy. So this is the angle, right? And this is the stretching. And then we run simulations for different choices, okay? So we tested different reversal frequencies. We would assume that each bacteria in all the simulations has the same length. And they would all move with the same velocity. We track them in experiment. They move with the same velocity for whatever reason. We don't know how they actually do that. But we realized that it's getting worse, right? I told you, bacteria is everywhere. Yeah. I don't know. We cannot go like this. Yeah.
Can you copy it to the desktop?
I can. I have it. I'll just keep talking for a while. So if you look at the system, it's not that different from a system of self-propelled rods. There's a huge literature on self-propelled rods. And also there's a huge literature on pneumatic crystals. So in this case, this bacteria moves on its own. There is no external field, right? And then they reverse, meaning that each cell has a clock. The clock is actually kind of set up by oscillating proteins, and I'll show, if I have a chance, to show movies on that. These are complicated systems, but that's the basic type of behavior is something very new to a physicist. Okay, so self-propelled rods have been studied a lot. Alignment was studied and so on. It's just this bacteria oscillates. So we tried to answer two questions. First of all, why it's beneficial for them to oscillate? And second, why they choose specific frequency to oscillate? And of course, this is a result of evolution. This is biology. They tried everything. So the diagram I was showing was kind of swarming rate versus reversal frequency. So we run simulations for populations of bacteria for different reversal frequencies. And for the frequency of 10 minutes, we got maximal swarming rate of the whole population. And that's exactly what population wants. It needs to spread as fast as possible because they need nutrient. So it's clear why it's beneficial, but what's the mechanism? So the problem with biologists, they were unable to track, even actually make bacteria fluoresce for a long time inside of a swarm because it's multi-layered and it's actually, this bacteria kind of slow. The movie I was trying to show was speeded up. So if you try to make them fluoresce, they actually die because any fluorescent dye, so I'm mixing two things, actually is toxic. Bacteria cannot survive for a long time. Thank you.
It's the wrong university for that to work.
This is what I'm accustomed to. So the answer is, and of course in simulations we can track them. Later we were able to come up with experimental setup, so we were able to track cells. We just improved this kind of resolution of the microscopy approach we're using. But basically this is what happens. If you track them in simulations, they jam all the time. It's like a traffic in Los Angeles or Chicago. I used to say Chicago, now it's Los Angeles. So when cars jam, there are two ways to get out of a traffic jam, right? Start moving slowly in the opposite direction, try to arrange better to not to jam again, or you can bend a little bit and just get out of it this way. It's exactly what this bacteria does. But the way it does it depends on the environment. You change environment a little bit, they're going to change the frequency. So the group at Berkeley actually produced mutants which actually reversed the direction of the motion at different frequencies. And they confirmed our prediction of the swarming rate. The point is, I can't keep talking. There's no excitement in that, right? This is mostly experimental work. Oh, okay, it's Kingston. Kingston. Yeah, that's the one. Yeah, I can take it from here. Yeah. Thank you. Thank you very much. Where did this come from? I don't need that. Okay. Okay, my talks. Yeah, okay. Here we go.
Install power.
This is a nightmare when you think about it. It's because I'm new on campus. First things first. Okay, very good. Mhm. Yeah, perfect, perfect. No movies still, but that's okay. So that's the diagram. So we run a lot of simulations for fixed frequencies. So all cells would have the same frequency of reversals. The phase would be different, distributed at random. And then we modeled 5,000 cells. And we looked at the swarm. And there's a way to kind of average out the edge. It's like a disk, and it's expanding in all possible directions. And 10, 12 minutes gave us the maximal swarming rate of the whole population. Then we changed the aspect ratio of a cell. The maxima shifted, but it was still there, because this is a result of evolution. So it's pretty amazing that, taking into account the complexity of what cells do, that they follow pretty straightforward kind of behavioral mechanism. So reversals, actually the driving force of getting out of traffic jams and maximizing the swarming rate. And what is even more interesting, if you look at proteins, the network inside of a cell, which is involved in reversals, and that's been tested by knockout gene experiments, actually the same components are used by other bacteria for chemotaxis. So chemotaxis, it's how cells, bacteria navigate. They just go towards maximum of chemical gradient. So 1 billion years ago, this bacteria decided to change the way of its life. It dropped the chemical navigation, rewired it for reversals. They started moving in big packs, like packs of wolves. They actually hunt other bacteria. They kind of attack a group of a different type of bacteria and go through them like butter. And these reversals help them to stay close to each other and self-organize in this very fast-moving groups of bacteria. Okay, so we talked to biologists, and they said, so big deal. You can just track bacteria and get the same result, or we can do it some other way. So we talked to Bustamante group at Berkeley. I think he moved recently to Stanford. And he came up with mutants. So he genetically modified bacteria, so different groups would reverse at different times. And we got a very good correspondence with the experiment. So we got confirmation. So we published a paper in PNAS, and then it was featured in Nature and later in The Scientist. And the title of the news article, Nature News, was, for bacteria to go forward, it needs to go backwards. Go figure. And now people are using similar, actually, behavioral algorithms to arrange groups of robots, actually, to do things together and move together. So we decided to improve the experiment. So we collaborated with Igor Aranson's group at Argonne National Lab. So we introduced this so-called imaging chamber. So we're able to put a drop of oil on the population and put a cover on it in such a way. It took a year. So bacteria wouldn't actually mutate, wouldn't change its behavior. And the outcome was very dramatic. This is how it used to look, and now how it looks now. We can actually track each bacteria in the population and confirm whatever predictions we have to make. Since we were able to look at and track each bacteria, we came up with a slightly more kind of complicated model. It's called subcellular element model. Basically, it's an extension, a coarse-grained extension of molecular dynamics. So each bacteria is still represented by certain number of nodes connected by springs, but then we introduce certain types of potentials from molecular dynamics to describe adhesivity between different bacteria. So this was implemented on graphical cards, clusters of GPUs, and it's very fast. We can model thousands of bacteria. At the same time, we were able actually to look at what happens inside of individual bacteria. So I talked about the kind of reversal mechanism. This reversal mechanism actually also is in charge of polarity of a cell. So self-propelled means that there is a head and there is a tail. So they move in specific direction. And how they decide about that is determined by certain proteins oscillating inside of a bacteria. And we decided to look at the division of bacteria and see what happens with this mechanism during division. And eventually, this is what happens. We were the first group to observe it because we were able to look at a very high resolution. So each bacteria during division stops. So it becomes more adhesive, adheres to the substrate. And then it grows to be twice the size. And other bacteria don't bump into it. Somehow they sense there is a bacteria there trying to divide. And I cannot show the movies for whatever reason. But other bacteria kind of approach dividing bacteria and start kind of bending and bypass it. They never push it. They don't interfere with division. But it's not exactly what we were interested in. We were interested in a simple question. After bacteria divides, what happens with daughter cells? What kind of polarity do they get? So what do you think? They get opposite polarity. They start moving away from each other. Why? Because they shouldn't be competing for nutrient. They're kind of relatives, right? It makes sense. But there is a mechanism which involves oscillating proteins, as I said, which is responsible for that. So just kind of, this diagram summarizes what happens. And the important thing is they move in the opposite direction. And we kind of confirmed it by tracking specific protein, which is called ROM-R. It's known to be involved in determining kind of oscillations. And it actually accumulates at the ends of a bacteria. But you see that it's also kind of is distributed all through the body of the bacteria. Depends on time. So what happens during division, it stops oscillating. Because bacteria loses polarity. It loses kind of identity, okay? And then they divide, and the big question is whether they become polarized after they already divide, or before they divide. And from our experiments for tracking proteins, we think that it first actually recovers polarity at the very first moment in time when the septum is formed, the separation in the middle of the bacteria. And then this polarity helps them, and forming motility mechanisms help them to push away from each other. So that's kind of a diagram of that. So it's a paper in Journal of Bacteriology. So now we have an extended model describing diffusion of proteins. We're trying to add more components we know actually control the reversals to the mechanism. But we're tracking proteins. We can track bacteria even better. So I want to show you how actually swarm of bacteria looks like. So there are layers. They self-organize in concentric rings. And this color indicates actually phase of the reversal frequency. They actually get, they kind of, in the end, they get close to other cells which have the same phase. So they start kind of oscillate together. And there's like space separation in that. And I think models from statistical mechanics can be applied to understand what's going on inside of a swarm. Okay. Now I'll talk about something completely different, as Monty Python used to say. I'll talk about other models, okay? And I'll try to explain what multi-scale means, at least for our group. So I talked about subcellular element model. Now I'll switch to a model which came from statistical mechanics, statistical physics. It's called cellular pods model. And it's an extension of the Ising, extension of the pods model. So I think people in this room know what Ising is. And Ising is a model of electromagnetic materials, right? So you have a lattice, and you have spins on the lattice pointing up and down. And then spins can flip, and they flip at random. That's the process describing dynamics of that model. And then you look at phase transitions, right? So, and of course, the initial Ising model is an equilibrium model. You're trying to run your simulation from point of view of kind of dynamics to get to the global minima of the system. So PLoS model is extension of Ising. Instead of just having spins or charges up and down, you make it multidimensional. Okay? And cellular PLoS model, extension of the PLoS model in the following way. Suppose we have distribution of multidimensional indices in such a way that you have clusters. Okay, so there's a cluster of nodes of type 1, type 3, type 2. And suppose there is a medium, which we're going to call extracellular matrix, and they present as particles of type 0. It's multidimensional because you can have several cells of the same type. So at this point, I made them all different, but they might be all kind of the same clusters. And the whole idea of the cellular PLoS model to run dynamics in a such way that these clusters move, and they can oscillate, fluctuate a little bit, but they wouldn't change their area and volume. So that's the Hamiltonian, which drives the whole process. So you have different clusters. You can calculate energy of adhesive interaction of nodes at the membrane. So for example, I have 1 and 2, right? So those two can interact, they're next door neighbors, and this interaction is going to contribute to the contact energy of the Hamiltonian. So the dynamics of this process, it's a Markov chain or stochastic process, which is a Markov chain. Basically, it's very similar to Ising. You flip, you choose at random a node and flip it. You change its state, okay? And then you calculate the change of energy. And if it reduces the energy, you accept it with probability 1. If not, this is Metropolis algorithm. The acceptance probability is a Gibbs measure. So you have a Boltzmann constant, you have temperature, which plays a different role in this specific model. So instead of having this as a temperature, this term determines fluctuation on the membrane of each individual cell. But you choose a node, you can flip it, and you can flip in such a way. Say you can flip, say 2 to 1, that this cell extends and this one shrinks. And you can repeat it again and again and again. Depends what happens with your energy, right? And then if you do this at random, your individual cells can grow to be huge, take the whole lattice, it's like phase transition, right? Or they can shrink to a point. So to avoid this, and don't forget this is a non-equilibrium model, we have this term here, which is a constraint. It's called volume term or area term. We have a target volume, and it's a quadratic. So if we overshoot the target volume a lot, this term becomes considerable. And next time you add additional pixel and increase this, it's going to actually increase delta E, but there is minus here. It's going to decrease the acceptance probability on the next step. If you try to make this cell too big, okay? Then it's going to be rejected, and so on. Okay, so people look at area and volume. And then you can take this cell and put in the external field. It's a different grid. It's separate. It's multi-scale. Now we're talking about multi-scale. So you need to calculate the chemical, it might be diffusing. Okay, and then that grid can be produced by a cell. And this is how cells actually interact through chemotaxis by producing chemicals and reacting to chemical gradients. But basically, this can increase probability of adding additional nodes at specific side of a cell. So cell starts kind of moving in that direction. And these are not real cells, right? It's a statistical mechanics model. It's a projection, okay? So it moves by adding pixels or nodes at one end and losing them on the other side. Okay. So for bacteria, different groups actually also added specific term which involves curvature to describe extended objects, extended cells. So that's additional term in the Hamiltonian. And this kind of models been and still are very popular. When people want to talk about the role of adhesivity between cells and adhesivity with the substrate. And cells are adhesive. And that's a big part of what they do. The other part is they actually also want to go through a random walk. If you leave them to that, they just move in a random direction. But as soon as they start collaborating or paying attention to each other, either they align with each other, start moving in parallel, or they start sensing chemical gradients, and so on. So I think that this model is not very good to describe mechanical stresses, for example, because it's a projection kind of on a screen moving. And the Hamiltonian also has terms which have parameters which are not very physical. So people were successful in calibrating those non-dimensional parameters by comparing with experiment, but these coefficients are non-physical. The subcellular element model is much more physical, and parameters there can be linked with the specific measurements, actually. Okay, so I gave an example of a statistical mechanics model, and it's multi-scale because we're coupling it with extended external field. I mentioned the chemical field, for example. We can look at this as a multi-scale problem also if you want to come up with mean field approximation. Okay, and I'll try to get through these calculations very fast. So it's not mathematics, it's physics. I'm not proving anything, right? That's the difference. Suppose we have a cell. Let's look at one-dimensional case. Cellular plots model, there is a cell on moving along a segment. That's the Hamiltonian. So we don't have volume, we have length. We have target length for this cell. And suppose we have a chemical gradient. So it moves towards maximum chemical gradient. We're going to look at the actual diffusion limit. Of course, we're taking into continuous limit, right? So far I was talking about physics. Now I'm going to look at, I'm a mathematical physicist, right? So I'm going to look at mathematical part of it. I like the differential equations. I don't know about you. Okay, I'll try to derive the differential equation and see whether it's right. It's a convenient way to describe behavior of the system. So I'll explain how we do it. So first of all, we write down the master equation. So we introduce probabilities of finding a cell of length L with center of mass at x at the moment in time t plus epsilon squared delta t. We're already introducing scaling for the time. And this is how we're going to do it, actually, for time and space, right? So in continuous limit, we're going to introduce more and more nodes in the lattice. And of course, we're going to switch to continuous time as well. And the ratio is supposed to be constant, right? It's a diffusion limit. This is how we're going to do this. So how do we get a cell at this center of mass here, length here, the next moment in time? Well, let's look back and see what might have happened, okay? So we might have had a cell which was slightly longer on one side, and then this node flipped into like 0, like being a part of the substrate. That's one outcome. So one of these constants, so it was plus. I need to find plus. I think that one. Okay, so that's a bit longer, and then I need to multiply by transitional probability. That's a probability of finding slightly longer cell close to this position at the initial moment in time delta t. Okay, and this is transitional probability of losing 1 pixel on the right-hand side. And how you calculate it? Using the Hamiltonian, it's delta E, right? That's exactly what, it's a Gibbs measure. And then it might have been slightly shorter and added 1 pixel, or it might have been slightly longer on the other end and lost it, and so on. Or nothing happened. It was there all the time. It was there on the previous step, and nothing happened. What probability of this happening? Transitional probability. It's 1 minus the sum of all the other transitional probabilities. All transitional probabilities are supposed to sum up to 1, right? Something should have happened. That's the equation for this specific kind of simple situation. Well, we go into continuous limit, and we came up with this partial differential equation. And it's called Fokker-Planck equation. And that was novel. Nobody else did it before us. And then we kind of were able to show that this equation has a specific type of solution, which we call ansatz. And I have a better explanation on the next step. So we can present a solution of the equation. Don't forget, we're talking about probabilities now, as a product of P of xl times P of xt. So we're separating distribution of lengths for our cells, for our cell, from actually position in space. So small p describes position of center of mass. It depends on x and t, right? And P of xl, it's kind of with a fixed position of the center of mass. You're talking about distribution of different types of lengths, or the different length distributions. So if you substitute it into the equation, you can show that for distribution of lengths, you just get Boltzmann distribution. And the equation for center of mass actually is just equation, so-called Keller-Siegel equation, the main equation in reaction diffusion equations, in the whole class of reaction diffusion systems, basically. So usually people use this equation without derivation, or they derive it by looking at randomly moving particles. It's a diffusion equation. But on top of it, we're talking about cells represented as rods. They have lengths. They have certain space characteristics for the first time, right? OK, and also we can couple it with the diffusion for the chemical, so it becomes reaction diffusion. The problem with the systems, they have blow up in finite time. Sounds very mathematical. Just imagine a bunch of particles moving on a semi-axis at random, right? So it's diffusion. And then suppose they're also coupled with a chemical gradient. And suppose the potential looks like this. So they all go to the center of this chemical potential. As a result, the density goes to infinity, basically. But it doesn't happen with these systems because they have finite size, actually. So when they get close to each other, the diffusion drops. So basically, what we get in this case is, well, this is a two-dimensional case for the same system. We get nonlinear diffusion. So diffusion coefficient, well, we also need to take into account exclusion volume principle. Diffusion coefficients depend on the density. And as soon as density goes up, the diffusion coefficient, it's 1 over p squared, actually drops. And this was derived for the dimensional case. So it's not just target length, it also has target, actually, width for bacteria. So that's the scaling for the new diffusion coefficient for this system. Why do we care for, say, partial differential equations? Who knows? Who cares? I care because I was trained that way. But basically, Cellular model, it's a stochastic system. It's Monte Carlo simulation, right? Is it enough just to run one simulation for that? It deals with probabilities. It's like running an experiment. You need to run this simulation many, many times and then average out. Takes a lot of time. Partial differential equation, you determine initial conditions, boundary conditions, solve it once, and that's it. The problem is there was a mismatch between modeling using partial differential equations and like systems of particles on the physics side. Because PDEs before were rarely derived from interacting particles. Or these derivations never took into account sizes of individual cells. Okay? So if you have a PDE, you get some terms, and these terms are not really physical or biological. And if biologists tell you that it's not what actually bacteria does, for example, you cannot change the PDE. You need to go to cellular POTS model, change your assumptions about behavior of the system, the Hamiltonian and acceptance probabilities, and derive equation again. So definitely PDE is much faster, right? But it's very, very rigid. So this whole field of mathematical biology, till maybe 15 years ago, was about partial differential equations, and it was sitting between, say, mathematics and mathematics. It's not just our group. A lot of people now derive continuous limits and try to justify different terms and PDEs from kind of certain assumptions about interaction between bacteria and cells in general. How much time do I have? 15 minutes? Okay. So 10?
I think we have to be out of here by 5.
15 minutes? Okay, good. So let me talk about slightly different example, biologically and model-wise. So this is Pseudomonas aeruginosa. So this bacteria, they have flagellum. So we run experiments in our own lab. We inoculate Petri dish with certain number of bacteria. Then we use a very sophisticated device. It's called toothpick. And you kind of put it there, put it in somewhere else, and put it there. It's like several million of bacteria. And they start growing. And initially just sitting there, because agar doesn't have any water on top of it. Agar, there is hard agar and soft agar, depending on how much water is there. But initially, it needs to be extracted. And this is what bacteria is doing. We're kind of observing it in the lab conditions, trying to figure out what is it they do in humans. Okay? So they have quorum sensing mechanism. They produce certain chemicals which accumulate on surface of individual cells and diffuse a little bit. So they know how many bacteria are in the neighborhood. And when the number hits specific threshold, there's a genetic switch in each bacteria. It's called quorum sensing. And they say, aha, we need to change our behavior. They start producing REM lipid. Why it's important for them to start doing something differently? What do you think? I mentioned this already. Maybe I'll give a quiz in the end.
Resource management?
That's right. Because if they stay too long in one place, there are too many of them, boy, they deplete everything. They start dying. They don't want to die. Okay? They don't mind dying for a reason. Okay? So I didn't tell you something about, say, Myxococcus hantus, because when you starve them, they start eating each other. Maybe I should have said that. I don't know. So bacteria is very sensitive to levels of nutrient. And if they're starving, they do something completely different. Okay? So this bacteria, after they hit specific density, they start producing REM lipid. And these REM lipids help them to coordinate the process of extracting water from the substrate. You see this water coming out of the substrate. So there's a blob of water going up. And then something very interesting happens. You see this bacteria. REM lipid also plays a role of surfactant. It changes the surface tension. So you see somewhere here on the boundary that imperfections on the surface of this blob of water get amplified by the drop in the surface tension controlled by surfactant, by REM lipids. So fingering occurs. A well-known phenomenon in physics, like this. And it's a well-known phenomenon. People usually demonstrate it by putting drop of oil on water. Okay? So it's called Marangoni force, which kind of reinforces the singularities or imperfections on the boundary. So why it's beneficial to the bacteria? So far, people have seen it. It has nothing to do with what we did. We were able to observe that after these fingers are formed, you see them over there. a ring is formed. This bacteria self-organizes in a ring, high-density ring, which starts very rapidly propagating towards the tips of the fingers. Okay? And then they populate them. And we call this secondary swarms, secondary populations, fingernails, for obvious reason. And the process repeats itself. So again, they stop, they divide. And then when they hit specific density, they start producing even more RAM lipid. And the second fingering occurs. You see the second fingers here. And they keep propagating. It's a very efficient way of taking over the surface. They don't know where the nutrient is. The water drives them. So at least one of the secondary populations is going to be in the area with high levels of nutrient. That's the hope. If you just let them propagate on their own, even if they swim, it's not enough. So it's not diffusion, right? It's something else. It involves Marangoni force, and they found a way to use this interaction and control of this microfluidic environment to their advantage. And of course, they optimized it. The levels of RAM lipid, how fast they move using flagellum, and so on. So we observed that, published the paper, but then we started thinking about mechanism. Modeling for us is the way to talk about mechanisms. Biologists actually are capable of modeling. They just don't know mathematics or physics at all. They just describe in a coherent way what they think the cell does to achieve certain outcome. They call it a model, all right? And if enough people believe in that, they publish it in Nature. And I'm not kidding. And it's viewed as a real progress. So modeling helps to check hundreds of different hypotheses, okay, and make it kind of persuasive enough so somebody's going to come up with experimental verification. So we came up with a model which actually combines thin film approximation of Navier stalks for very thin biofilm. And then we actually track motion of individual bacteria, 100,000 of them. And there are different parts of the simulation. So there's a precursor film and cutoff. And this is where we study carefully the Marangoni effect. And that's the equation describing the height of the biofilm. And then the Marangoni-driven instability is indicated in red. And we were able actually, and the way we modeled bacteria is very similar to what we did with the previous bacteria. So there's like several nodes connected by springs. They can bend, they can scratch, and there's a stochastic component and so on. But they're self-propelled. They have flagellum. So we made the assumption about alignment. We assume then when they get close enough and we have a characteristic distance between bacteria, they actually stay aligned for some time. We kind of favored that in our simulation. Only with that assumption, that was a model prediction, we're able to get a concentric ring propagating with a specific velocity, which was observed in experiment. So that was a prediction. After that, biologists, and we participate in experiments, that's a paper in Biophysical Journal, we improved the resolution of the experiment and calculated level of alignment. That's the parameter we used, the alignment, in the fingers and inside of the swarm. And we actually got very close to confirming what the prediction was. The level of alignment predicted in simulations was confirmed in experiment. So this is experimental snapshot of experimental movie. These are levels of water, and I think this is water. This is concentration of Pseudomonas, density of bacteria. Okay. So alignment is very important to them. This is the mechanism. They align in order to move much faster, which was not known in bacteriology before that. And then we decided to look at the mechanism, how do they align? And these bacteria have pili. Remember I talked about Myxococcus hantus. They have pili only in front. This bacteria has it all over the body. It's kind of hanging there. And biologists never thought it was helpful. They thought it's just something, it's just hairy bacteria, okay? So it's a question of fashion, I don't know. So we thought otherwise. We thought, well, probably when they get close to each other, they don't use them actively, but they get entangled with each other. And they have characteristic widths, and actually they don't allow bacteria to get too close to another bacteria. So we tried to test in simulations kind of different ways of bacteria kind of being close to each other. At the same time, we decided to come up with mutants. So this is biology for you. Biologists do two types of experiments. Molecular signaling, they're measuring levels of molecular interaction, and then genetic manipulation. And we've done genetic manipulation. We came up with a bacteria which didn't have any hair. Probably was very embarrassing to that bacteria. But they swarm like crazy. They get close to each other. We call them super swarmers. That's the one. That's the normal bacteria, the hairy one, super swarmers. We looked at a variety of mutants which had some presence of this PLi4, and we kind of looked whether they aligned better or didn't align too well. So this is super swarmer. They just align in the perfect way, get really close to each other, move very fast. This bacteria kind of gets entangled. It slows them down and so on. We mix them together and realized that whoops, I don't have this picture. We realized that super swarmers have an advantage. Since they don't care about others, they don't entangle. They get to the edge very fast. So they're first to come to get the food, because that's where food is. Okay, so life so good for them. Why we don't see them in nature? So we decided to expose our population, super swarmers and actually wild type, to an antibiotic. And the super swarmers just go all the way. They jump into antibiotic and die. And the wild type, the wise ones, they actually stop and use this pili, like hands, they're holding hands, they stop. That's the way they stop. So being slow is actually important when they get exposed to a poison. Don't rush into it, okay? And the way bacteria does it, they have a genome. They've been around for 3 billion years, this specific bacteria. They lived on Earth when life was real tough. We cannot even imagine how tough it is. So this is what bacteria does. After they get exposed to toxic material or whatever, antibiotic, they think, wait, I've seen it before. I've been exposed to poisons. It scans the genome, finds the right one, and kind of starts behaving that way. They start actively using pili, which was absolutely amazing. But microbiologists didn't have a clue that would happen. Of course, they also start producing certain chemicals, signaling to each other to do that. And we're trying to measure levels of the signaling at this point. But this active kind of way of using pili, it's a biophysical effect, okay? And we kind of measured the level to which they use that specific mechanism. And we're also studying them in microfluidic devices. So it's a collaboration with University of Chicago School of Medicine, surgery department. So we take epithelium cells, put on the substrate, and then we kind of mix populations of Pseudomonas, wild type, with the ones which were, we get them through a rat. We get them kind of irritated, okay? And then the ones which get irritated, we use antibiotic for that as well. They actually immediately go to the epithelium cells. And we use laser to create very small injury. And aggravated Pseudomonas goes to the injury site and goes through it, interferes with wound healing. So that's the real danger of this infection. people after surgeries do not heal and die, which is a big deal. And when you hear about bacteria becoming antibiotic resistant, that's the one they're talking about. Pseudomonas is it's one of them. It's like 4 times drug resistant. Okay, you cannot kill it because it loves antibiotics and the rest of it. Okay, so let's see if I can run this movie. And I can't. All right. So the last project I want to mention is blood clotting. So we all have blood, right? And we kind of function properly. What we don't appreciate that it's a very delicate kind of system. It's very sensitive. And if something goes wrong, the outcomes are kind of immediate. So I mentioned that blood clotting is a leading cause of death in the United States, and it gets overlooked because people get clots and die of them when they have other conditions. Obesity actually contributes to formation of blood clots. Any irregular heartbeat creates blood clots, very small ones. They shoot up to brain and people get paralyzed. People with diabetes form blood clots. People with cancer, many of people with cancer actually survive cancer, die of blood clot forming in the middle of the night. Because all the medications used change the biochemistry of blood, but also blood is not just liquid, it's plasma with large number of different types of cells. And we are studying what happens during clotting with different types of cells. And the main of them is called platelets. These are the cells which get activated in case of emergency. So here you're looking at a very small blood clot forming in the blood vessel ear of a mice. We use laser, creating small injury. And kind of injured epithelium cells produce certain chemicals which go into bloodstream. And platelets get activated. So in our experiments, they become green. They fluoresce, activated platelets fluoresce, and they become very, very adhesive. And they change their shapes. They're usually like disc shaped, they become star shaped, become very adhesive, they stick together. You see formation of clusters, and some of these clusters adhere to the injury site. It's a plug. So any kind of injury results in internal bleeding, which is deadly. This is why every person after a car accident is supposed to be taken to hospital for checkups, internal bleeding. So we have a mechanism which actually deals with it if the injury is not too big. Okay? And then, which is very interesting, activated platelets start coagulation pathway reactions in blood, and fibrinogen present in blood polymerizes into a fibrin network, which kind of cements the clot. Otherwise, the clot is not going to survive the pressure, sustained pressure from the flow. It's a porous medium, okay? And in healthy individuals, it all comes together in an optimal way, and this clot doesn't grow to be too big or too small, okay? Nowadays, unfortunately, they can grow to be too big, they clog the artery, and people die. Or they become very irregular, get detached, end up in lungs, and people die immediately. So we study using cellular pods model, how these clots can form. And it's a multi-scale model. So on one lattice, we have this cellular pods model representing cells, different types of cells. These ones actually have already fibrin network covering them. It's activated platelets, non-activated platelets, other bigger blood cells, non-adhesive. And then we have a Navier-Stokes or particle flow representing blood. And then we look at the interaction, and we go back and forth in space and in actually time. So we run simulation for cellular pods for a while, just taking all the kind of measurements from the simulation of the fluid, applying pressure on the boundaries and deforming them. And then, after this is readjusted, we run code for Navier-Stokes and see what happens with the flow. So we made certain predictions, and this is one of them. for specific shapes of clots, actually we see formation of the verticity. And this vertex is actually pushing inside of the clot larger blood cells which are not adhesive at all. And before we started writing about that and publishing, people always were focusing on just platelets. And now in medical schools, people are actually looking at clots and realizing that a big part of what happens inside of the clot is just larger non-adhesive cells, which actually compromise mechanical stability of the clot. And all medications on the market target at this point the adhesivity of platelets and nothing else. And there are a lot of clots which don't have too many platelets, mostly fiber network, especially the ones which cause strokes. And we're looking at studying fiber networks as well. That's a simulation, subcellular element simulation of two cells in blood, 3D simulation, kind of adhering to each other. And this is a simulation of a platelet adhering to the substrate treated with specific components found on the surface of a blood clot. This is a actually experimental movie. This is how we calibrate the model. And kind of moving. So you can see individual platelets moving and actually touching the substrate. So it's not like they are rolling, they are flipping. So they're very adhesive and they have specific receptors which can be attached to specific ligands on the surface. Okay, so it kind of moves a little bit, stops, moves again. It's a very delicate procedure. But this needs to be studied in experiment and simulation in order to come up with new ideas for drugs, which would prevent platelets from attaching to existing blood clot. Otherwise, the clot grows too big. And people with this kind of clots, they have certain genetic deficiencies. There are a lot of kids being born with certain deficiencies which result in formation of these very large clots. So just we also study fiber networks. We use confocal microscopy, reconstruct three-dimensional structures and characterize them. So we usually started by activating one platelet, and then it kind of results in polymerization of the network. And then what's interesting, platelets kind of live in this environment. They communicate by pulling different fibers in the fiber network. And if everything goes right, at the last moment after formation of a clot, they all, in very coordinated manner, pull on fiber network. It's like having a parachute. And the whole structure actually becomes very compact. But it becomes compact and perfect for specific flow rate. If something goes wrong, that becomes very dangerous. These are my collaborators. So on Bacterial project at Josh Shroud from Notre Dame, that's my former postdoc, Cameron Harvey. So we collaborated also with Igor Aronson from Argonne on experimental side. Denny Chen is a computer scientist who does image analysis. These are my former students, and my collaborator, Zhileng Shu from Notre Dame. This is our funding, mostly NIH. And these are collaborators on the blood clot project. In addition, I want to mention Rustam Litvinov and John Weisel. They are from University of Pennsylvania School of Medicine. They're leading experts on fiber networks in clots. And we're funded by NIH U01 for studying blood clotting using multi-scale modeling. And thank you for your time. Well, they're self-propelled. So they basically kind of, when they push each other, they tend to align. So it's a self-propelled rod. Well, at the same time, they're covered in slime. So when they get aligned, they stay aligned, at least for a while.
So it's like they get more space this way by alignment? Liquid crystal alignment?
So don't forget, they also, they can reverse. So if you have like two groups going like this, and they jam, they stay aligned, but they They're trying to resolve the traffic jam that way. And alignment helps them to do that.
And then they do swarming because
So swarming, it's a loose term describing any kind of social behavior of individuals. In this case, they swarm because they're self-propelled, and then because they're social. And don't forget that for Myxococcus hunters, they also have pili in front of them or head of them. So they kind of test the other bacteria. They try to follow other bacteria as well. But
in bears they say there is like magnetic field or something, magnetic field following this.
not for them.
Not-
They're individuals, but they do things locally. But this local kind of algorithms or rules they use help the whole population to swarm at maximal rate.
Is the flow turbulent?
No. In which problem?
Well, when you're talking about the clotting, if you had flow, whenever you had the nebulous spokes in there, and you were mentioning there were vortices and the importance of the vortices. Now, are the vortices, they're not a result of turbulent flow, that's all laminar?
There are cases when it's actually not just laminal, because some clots are formed and they have two maxima, so they might have something in the middle.
So it's all in the laminar regimes?
Not necessarily.
Not
Not necessarily.
That's not even an issue. It can be an either, or?
It's been studied, actually. Don't forget, it also pulsates flow. It's pulsating. So it depends where it
Well,
forms.
that's what yeah, so
Yeah.
I mean, there are obviously advantages to having a pulse flow in the circulatory system.
Right.
And what I'm not sure of is whether or not there's advantages to having turbulent flow in the circulatory system for the same reason.
So usually it's not good.
Turbulent flow.
Yeah.
I
But
know it's not good for an airplane, but
but
when you're talking about getting things into really small cracks,
I didn't mention another kind of situation when clots are formed. Any kind of implant results in blood clotting. So any kind of corrections of the heart might result in turbulent flow, and they start clotting like crazy. So it's usually people look at different surfaces and their shapes.